I am Vijay Durga, working as a botany lecturer in Sir Sierra College for Women, Eluru. I am dealing with the paper cell biology, genetics, and the plant breeding. Today, I am going to discuss about the topic Mendel's laws of inheritance. Mendel's laws of inheritance. Mendel is a scientist. He is botanist and he is an American monk. He introduced the process how the genetic material is passing from generation to the generations and how the heredity works. He has conducted experiments on the Pisum sativum, that means in the garden pea plants. He has taken and worked out and conducted experiments and proposed a mainly laws. Inheritance can be defined as a process how a child receives genetic information from the parent. That means the genetic material is passing from one generation to the next generations. Why? Means during the gametogenesis process, the parent 50% of genetic material is generated to the next offsprings. That means they are getting the same genetic material which possesses similar characters. The whole process of heredity is depend upon the inheritance and it is the reason that the offsprings are similar to the parents. In our family we are seeing that children are similar to the parents. Why? Because the genetic material is present in them are identical, which means they are similar genetic material possessing. That's why only they possess similar characters. This process is called inheritance. This simply means that due to inheritance, the members of the same family possesses similar characters. అంటే మన ఇంట్లో మనం చూస్తూ ఉంటాం ఒక ఫ్యామిలీలో ఉన్న మెంబర్స్ అందరూ ఒక సిమిలర్ క్యారెక్టర్స్తో ఉంటారు. వై బికాస్ మీన్స్ డ్యూ టు దిస్ ఇన్హెరిటెన్స్ ప్రాసెస్ ఓన్లీ ద జెనెటిక్ మెటీరియల్ ఈస్ పాసింగ్ ఫ్రమ్ జనరేషన్ ఆఫ్టర్ generations and possesses similar characters. It was only during the mid 19th century that people started to understand the inheritance in the proper way. మిడ్ 19th సెంచరీ నుంచే పీపుల్కి అవేర్నెస్ క్రియేట్ అయింది. ఏమిటి అంటే దట్ జెనెటిక్ మెటీరియల్ అనేది సేమ్గా ఉండడం వల్లే పీపుల్ అనేవాళ్ళు ఒక సేమ్ సిమిలర్ క్యారెక్టర్స్తో ఉండడం జరుగుతుంది. దిస్ ఇస్ ఓన్లీ డ్యూరింగ్ ది మిడ్ 19th century. It has started from mid 19th century that people started to understand the inheritance in the proper way. This understanding of inheritance was made possible by the scientist named Gregor Mendel. He is only the person who creates awareness about the inheritance, who formulated certain laws to understand the inheritance known as the Mendel's laws of inheritance. He has conducted experiments on the pea plants and studied about the generations, and he has formulated certain laws to understand the inheritance process, the passing of genetic material from generations to the generations. He has proposed these Mendel's laws of inheritance. Between 1856 to 1863, Mendel conducted the hybridization experiments on the garden peas. He has grown the garden pea plants, that means Pisum sativum he has taken and the grown he is maintained easily and he has done artificial or cross self-pollination techniques and he has undergone the hybridization techniques and conducted experiments on the garden pea plants. During that period, he chose some distinct characteristics of a piece and conducted some cross pollinations as well as artificial pollinations on the pea lines that showed stable trait inheritance and underwent a continuous self pollinations. During the process of experiment, he has understand that the passing of genetic material. Why means generations after generations the genetic material is passing in the stable way. That means the offsprings are processing it that similar characters they possess. That's why he has understand that. Why means the inheritance process, that means heredity, genetic material, how the genetic material is passing from parent to offspring generations. By conducting these experiments only he has understand that the stable trait inheritance and underwent continuous self-pollination. Such pea lines are called the true breeding pea lines. ఆ పీ లైన్స్ని ట్రూ బ్రీడింగ్ పీ లైన్స్ అంటున్నాం. Mendel. Why was pea plant selected for the Mendel's experiments? మెండల్ ఎందుకని పీ ప్లాంట్ని సెలెక్ట్ చేసుకున్నాడు, వేరే ఏదైనా అదర్ ప్లాంట్ని సెలెక్ట్ చేసుకోవచ్చు కదా అన్న డౌట్ వస్తుంది. He has given some reasons why he has selected the pea plant. That means he selected the pea plant for his experiments. The pea plant can be easily grown and maintained. It can be grown easily and it can be maintained easily. అంటే మనం దాన్ని గ్రో చేయడానికి అంత దట్ మచ్ కేర్ తీసుకోకర్లా మనం. అంటే ఇట్ కెన్ బి గ్రోన్ మెయింటైన్డ్. దే ఆర్ న్యాచురల్లీ సెల్ఫ్ పాలినేటెడ్. అంటే దానికి అది పాలినేషన్ జరుపుకుంటుంది. But it can also be, if we want to do cross pollination techniques, we can also do cross pollination techniques. It's an annual plant. That means its lifespan is going to be completed in the only 1 year. Therefore, many generations can be studied within the short period of the time. అంటే వేరే ఏదైనా ప్లాంట్ తీసుకుంటే మనం దాని యొక్క డేటాని కలెక్ట్ చేయడానికి మనం మెనీ జనరేషన్స్ అనేవి స్టడీ చేయాల్సి వస్తుంది. కానీ ఈ పీ ప్లాంట్ని తీసుకోవడం వల్ల ఏంటంటే దాని యొక్క లైఫ్ స్పాన్, అంటే దాని జీవిత కాలం అనేది ఓన్లీ 1 yearలో కంప్లీట్ అవుతుంది. కాబట్టి మనం మెండల్ అనే అతను సైంటిస్ట్ ఈ పీ ప్లాంట్ని సెలెక్ట్ చేసుకున్నాడు. అంటే మెనీ జనరేషన్స్ యొక్క డేటాని వితిన్ 1 yearలో ఈజ్ కలెక్టింగ్. వితిన్ 1 yearలో తను కలెక్ట్ చేసుకుంటున్నాడు. దట్స్ వై హ్యాస్ టేకెన్ పైసమ్ సెటైవం ప్లాంట్. It has several contrasting characters. That means the pea plant is showing 7 different types of contrasting characters. That means height of the plant, flower color, flower position, pod shape, pod color, etc. Mendel conducted 2 main experiments to determine the laws of inheritance. These experiments were, he has taken that garden pea plants, they grown and done pollination techniques. He has done experiments to determine the process of inheritance or the heredity process. He has conducted mainly 2 experiments to propose the laws. They are monohybrid cross experiment and the dihybrid cross experiment. During the process of experimenting, Mendel found that certain factors were always being transferred down to the offsprings in the stable way. That means he has observed that the passing of genetic material from parent to the offspring generations, the genes are transferred in the stable way without any changes. Why he has observed means the offsprings are possessing similar characters. అంటే మన యొక్క offsprings అవి similar charactersతో ప్రొడ్యూస్ అవుతున్నాయి. That's why he has understand that process of inheritance and the genes, the transfer of genes, he has understand. Those factors are now called genes. Genetic material అనేది పాస్ అవుతూ వస్తుంది generation after generation. He called these genetic material as genes. The genes which are present in this genetic material is responsible for the inheritance. And Mendel called these genes called as a factors. Those factors are now called genes. Genes can be called the unit of inheritance. These genes అనేవి యూనిట్ ఆఫ్, అండ్ దట్ మీన్స్ ఇన్హెరిటెన్స్ ప్రాసెస్కి ఈ జీన్స్ అనేవి రెస్పాన్సిబుల్ అని మెండల్ మనకి ఎక్స్ప్లెయిన్ చేస్తూ వస్తున్నారు. Mendel's experiments. I discussed already about that 7 contrasting characters of the plant, and he has conducted the experiments on the pea plants to propose the laws. Mainly he has conducted 2 main experiments, that is monohybrid cross experiment and the dihybrid cross experiment. In the next slide we are going to discuss about monohybrid cross experiment. In this monohybrid cross experiment, what happens means, here he has taken the 2 traits, that means tall plant and the dwarf plant. He has taken here the tall plant is written as a, this is the gene. In the one gene we are having the 2 alleles, that means here which is having the 2 alleles, that is dominant. Here it is the dominant. Capitalization letters are the dominant in conditions. These are the small letters which are in the recessive conditions, which means these are the dominant genes are the suppressed by, these are the suppressive in conditions, which means recessives. This is the tall plant and this is the dwarf plant. These are the parents generations. In this experiment, Mendel took 2 plants of the opposite traits, that means one short plant and one tall plant. and crossed them. He has crossed them. He found the first generation offsprings were tall and called it F1 progeny. That means he has taken these parental generations and crossed these two plants. One tall plant, one dwarf plant. He has crossed it, and we get one offsprings. That is F1. We call this hybrid as a F1 hybrid, and we called it as a offsprings. That is F1. These are the gametes, and we combine this. We get the F1 hybrid. If we called it as a F1, and he found the first generation offsprings were tall and called it F1. This is the F1 progeny. Then he crossed the F1 progeny and obtained both tall and the short plants in the ratio 3 is to 1 ratio. It means he has taken this F1 hybrid and he has crossed self pollination. Yes, these offsprings are pollinated by itself and has self pollinated. It has undergone self pollinations and written in the checkerboard. One one parent one parent gametes are written on one side and the next gametes are written on the next side. When these are combined, we get the homozygous tall, dominant homozygous tall. This is the dominant and recessive. We get the heterozygous tall. That means we are having two different types of alleles, capital T and the small t. In this, we are going that here the capital T and the small t are combining. That means heterozygous tall. Capital T1 and small t1. These are different. That means heterozygous tall. Here it is the homozygous dwarf. That means both are in the recessive condition. That are homozygous dwarf. This is the monohybrid cross ratio. Here the phenotypic ratio. Phenotypic means the characters which we are seen from outside. The tall or the dwarf. We are seeing that three tall plants, tall, and the tall. We written as 3 is to 1. That means homozygous dwarf. This is 1. That 3 tall and 1 dwarf plants. Genotype means based on the allelic conditions, the genotypes are written. Here the homozygous, that means capital T, capital T. This is 1. And here we are seeing the two different types. Capital T, small t, capital T, small t. 2. 2 is to 1. That means homozygous dwarf, small t, small t. That the genotypic ratio is 1 is to 2 is to 1. Okay, clear? And we got the ratio that 3 is to 1 in the phenotypic ratio. That means 3 tall plants and 1 dwarf plant. Mendel even conducted these experiments with other contrasting traits like green peas. Like this tall plant and dwarf plant, he has taken green peas versus yellow peas and the round versus and winkled, etc. In all the cases, he found that the results were similar. From this, he has formulated why he has done these experiments with other characters also, like green peas versus yellow peas and the round versus twinkle, and he has found that the results were similar. Whatever the results we get that are similar. From this he has formulated that laws. That means laws of segregation and the laws of dominance. He has proposed these laws. Laws of segregation and the law of dominance he has taken. The Dihybrid Cross experiments. It is in this he has taken two contrasting characters. If it is taken one contrasting character, it is having two alleles. In the this it is taking two contrasting characters. That means it is having the four alleles. That means four different types of alleles are having. In the dihybrid cross experiment, Mendel considered two traits, each having two alleles. uh, Mendel has taken two traits. That means each trait is having the two alleles. That means in one contrasting character it is having the four alleles. He crossed the wrinkled green seeds and the round yellow seeds and observed that all the first generation progeny were round yellow. In this slide we can see that he has taken yellow round seeds, this and green wrinkled seeds. These are the parental. Here this is the contrasting characters. This is one, that means yellow, capital Y, round, capital R. This is green, small y. This is smaller, that means wrinkled. When these are the combined, we get a capital Y, small y, and the capital R and the small r. These are the four. This is the F1. We get the offsprings. This is the F1. When it has undergone the self-pollination technique, we get the more, more amount of yellow round seeds we got. yellow seeds and round seeds, and they observed that all the first generation progeny were yellow round seeds. This meant that the dominant traits were the round shape and the yellow colors. Here we are seeing the dominant trait. This is the dominant trait. That means yellow round colors. These two are responsible for the yellow, and these two are responsible for the round. This means which possesses yellow round color. This means the dominant traits. He then self-pollinated the F1 progeny and obtained four different traits: wrinkled yellow, round yellow, wrinkled green seeds, and the ground green in the ratio 9 is to 3 is to 3 is to 1. In this checkerboard, we are seeing that we get after this after going the self pollination or the self fertilization technique, we get the four different types of the traits, like yellow round seeds and the green round seeds. This is yellow wrinkled seeds, and this is the green wrinkled seeds. We get the four different types of the traits after undergoing this self pollination. The whole process gives these rise to these four different types of the characters. After conducting for the other traits, the results were found to be the similar. He has conducted and the different experiments on the other contrasting characters. He was observed the same result. From this experiment, Mendel formulated his second law of inheritance, that is, law of independent assortment. Based on the monohybrid cross experiment, he has proposed the laws of segregation and the law of dominance. Based on this dihybrid cross experiment, he has proposed the law of independent assortment. In this checkerboard, I am giving now detailed view of this checkerboard. These are the parental generations which undergone the when these are the combined, we get the F1 progeny, and when this F1 progeny undergoes the self pollinations, we get the four different types of the gametes. These are the four different types of the gametes. Four different types of gamete from the one parent are written on the one zygote. That means gametes from the heterozygous parent, and this is from the gametes from the heterozygous parent. These are written on the one side. When these are the combined, that means capital Y, capital Y, capital R, capital R. If it is having the capital Y and the capital R, it is possessing the yellow round seeds. In this we can see capital Y and the capital Y. It is possessing yellow round. Capital Y, capital R, yellow round. It is also having capital Y, capital R, this yellow round. Why? Because means presence of the dominant alleles. The capital Y and the capital R are the dominant in condition. That's why they possess their phenotype is the yellow round seeds. But here it is having the recessive capital Y, small y, and the capital R, capital R. That means due to this small y, this is in the suppressive condition. It possesses green color. These are in the dominant condition. Possesses round shape. Yellow round is having capital Y and capital Y. It is also same having small y is the green round. This is also capital Y, capital R, it's having yellow round, and this is the capital I, capital R, this is yellow round. This is the capital Y, and the small r and small r. This is the yellow wrinkled, and this is also yellow wrinkled. This is the capital Y and the capital R. This is the yellow round. Small y is present, that is green round. It is capital Y. It's only the capital Y. That means it only possesses yellow color, and the recessive smaller and smaller. Yellow wrinkled. It is both are recessive. That means both the traits. That means small y and the smaller are recessive. These are double recessive. This possesses green wrinkled. These four different types of traits are the produced by this undergoing the self pollination technique. In the dihybrid test cross ratios, in the F1 hybrid plant, round yellow piece is crossed with the homozygous double recessive genotype wrinkled green piece. Here the round yellow cotyledons are crossed with the green cotyledons. This is called the dihybrid test cross with a ratio of 1 is to 1 is to 1. Here the yellow cotyledon, capital R small r and the capital Y and small y. Here the small r and the small y. This means yellow cotyledon is crossed with the green cotyledon. Gives rise to gametes. These are the four different types of gametes. When these undergoes the self pollinations, we get test cross, means either of the parent he has crossed this, and we got the ratio in the 1 is to 1 is to 1 is to 1 ratio. Mendel's laws. Based on this monohybrid cross experiment and the dihybrid cross experiment, he has proposed mainly three laws. The two experiments lead to the formulation of Mendel laws known as laws of inheritance, which are called. This law of dominance and law of segregation are proposed by the experimenting of the monohybrid cross experiment. The law of independent assortment proposed by the based on the law of dihybrid cross experiment. First law is law of dominance. This is also called the Mendel's first law of inheritance. According to this law of dominance, hybrid offsprings will only inherit the dominant trait in the phenotype. The alleles that are suppressed are called as the recessive traits, while the alleles that determine the trait are known as the dominant traits. Here, Mendel has proposed that according to this law of dominance, dominance means capitalization letters that are in the dominant conditions. Their phenotype is going to be expressive. Why? Other character is the recessive in condition. That means their alleles are in the suppressive condition. If we take in the tall plant and the dwarf plant, tall plant is written with capital T, and the dwarf plant is written with smaller and small t and small t. The capital T and capital T are dominant. Why? Because it's having dominant alleles. They are dominant. They are expressing their phenotype. And the small t and small t are in the suppressive condition. That means that they are in the recessive condition. They possess recessive. They do not show their phenotype. Here he is showing that hybrid offsprings will only inherit the dominant trait in the phenotype. The alleles that are suppressed are called the recessive trait, while the alleles that determine the trait are known as a dominant trait. That means the alleles which are showing their phenotype trait are known as a dominant traits. Law of segregation. In this, this means law of segregating. In this word only we can understand that segregating means combining. In this law of segregation, the law of segregation states that during the production of gametes, two copies of each heredity factor segregate so that offspring acquires one factor from each parent. In other words, or allele, pairs segregate during the formation of gamete and reunite randomly during fertilization. This is also known as Mendel's third law of inheritance. Law of segregation means in this process, during the gametogenesis process, during the formation of gametes, the genetic material is going to be produced from the parent to offsprings. During that process, the father possesses 50% of genetic material and the mother can share 50% of genetic material. During these fertilization process, what happens means the genetic material from the both the parents are segregating with each other. That means they are mixing their genetic material and the combining their genetic material with each other. During that process, the characters, the genes responsible for the characters, in the genes we are having the alleles. These alleles are combining with each other. That means the alleles are transferring. Due to that process, the phenotypic expression is going to be changed. This is only the law of segregation process. Law of independent assortment. That means the alleles, whatever the genes that are responsible for the phenotypic expressions, the genes, in the genes we are having the alleles. The alleles are assorted independently. That means during the gametogenesis process, there is no mixing of genetic material, there is no combining of genetic materials. There is no segregations. That means the alleles are sharing without in the independent assortment process. There is no segregation process. The law of independent assortment, also known as Mendel's second law of inheritance. This is also called as Mendel's second law of inheritance. The law of independent assortment states that a pair of a trait segregate independently of one another pair during the gamete formations. As the individual heredity factors assort independently, different traits get equal opportunity to occur together. During that process, heredity process, the passing of genetic material from one generation to the next generation is passing. During that process, the alleles are transferring that are assorted independently without any segregation process. This is only the law of independent assortment. Backcross. Backcross means it is the crossing of a hybrid with one of its parents. We can see in this plant, this is the pure tall plant and this is the pure dwarf plant. In this genotype, capital T, capital T. These are the alleles. This is the genes responsible for the trait. This is the these are the recessive genes which is responsible for the dwarf conditions. When these are crossed, we get the one F1 hybrid. But in this backcross method, we are doing this. This F1 hybrid is crossing with either of the parent, either tall plant or either dwarf plant. Here we are seeing that in the checkerboard that this is the F1 hybrid, capital T, small t. We are crossing this with the homozygous tall plant. That means it is having the homozygous means same alleles. Homo means same. Hetero, this is the heterozygous and means different alleles. When these are crossed, we get the two tall, whole are tall plants. But when we see in the genetic ratio, that means when we see in the difference in the alleles, that is capital T small t, capital T small t. Here we are seeing that two different types of genetic genotypic materials. We are seeing that capital T is one and capital T small t is one, that different types. This is a crossing of hybrid with one of its parents or an individual genetically similar to its parents in order to achieve offsprings with a genetic identity which is closer to that of a parent. It is used in the horticulture, animal breeding, and in the production of gene knockout organisms. In this backcross method, the hybrid offsprings, that means F1 hybrid is mostly crossing with either of the parent, that means dominant or it may be the recessive, or if it is not, it can be crossed with a genetically identical parent. That means the genetic material should be in the identity, that means they should have identical genetic material, their genotype must be same. That's why he has crossed that F1 with a genetically identical parent. Mostly this backcross method is used in the animal breeding process and in the production of gene knockout organisms, mostly used in the horticulture. The backcrossed hybrids are sometimes described with acronym BC. The backcross is sometimes describe మనం BC అని డిస్క్రైబ్ చేస్తాం అనమాట. ఫర్ ఎగ్జాంపుల్ an F1 hybrid crossed with one of its parents or the genetically similar individual. బ్యాక్క్రాస్ అంటే మనం ఒక పేరెంట్ ఐదర్ ఆఫ్ ది పేరెంట్తో కానీ లేకపోతే జెనెటిక్ ఐడెంటిటీ ఉన్న ఇండివిజువల్ ఆర్ ది జెనెటికల్ సిమిలర్ క్యారెక్టర్స్ ఉన్న ఒక ఇండివిజువల్తో కానీ క్రాస్ చేస్తాం. F1 హైబ్రిడ్ మళ్ళీ నెక్స్ట్ క్రాస్ చేసిన తర్వాత మనం దాన్ని BC1 అంటున్నాం. దట్ మీన్స్ బ్యాక్ క్రాస్ 1 హైబ్రిడ్ టు ది సేమ్ పేరెంట్ ఆర్ ది జెనెటికల్లీ సిమిలర్ ఇండివిజువల్స్. ఇట్ processes BC2. That means F2 means F2. BC2 hybrid it can possess. In the test cross ratio, in this test cross ratio, in genetics, a test cross first introduced by the Gregor Mendel involves the breeding of an individual with a phenotypically recessive individual in order to determine the zygosity of the former by analyzing the portions of offspring's phenotype. That means in this test cross, the F1, in the back cross, we are crossing the F1 hybrid with either of the parent, either dominant or it may be the recessive. But in these test cross conditions, we are crossing the F1 hybrid with only with the recessive. In this cross, we have crossed this F1 hybrid with 12 plant. But in the test cross ratios, we are crossing this F1 hybrid with recessive alleles. We are crossing these recessive, which are in the dwarf conditions. The traits are need to be crossed in the test cross. Phenotypically recessive individual. In order to determine, mostly this test cross is used for the zygosity of the farmer. To know the phenotype of the plants, we are using these test cross to determine the zygosity. Zygosity can either be heterozygous or the homozygous. Those that are heterozygous have one dominant and one recessive alleles. I already said that hetero means different. It's having the two different types of alleles. That's why it's processing different types of the character. Homo means same. It is having the same type of alleles. Individuals that are homozygous dominant have two dominant alleles, and those that are homozygous recessive have two recessive alleles. Homozygous dominant have two dominant alleles, and those that are homozygous recessive have two recessive alleles. Homozygous dominant means both are in the capitalization form, both are in the same condition, capital T, capital T. And the homozygous recessive means this is also same. Both are in the same and these are in the recessive condition, that are the homozygous recessive have two recessive alleles. Key points on Mendel's laws. The law of inheritance was proposed by the Gregor Mendel after conducting experiments on the pea plants for the several years. He has taken the pea plant, conducted a cross pollination and artificial pollination techniques and hybridization techniques proposed mainly three laws, that is law of dominance, law of independent assortment and the law of segregation he has proposed. The Mendel's laws of inheritance includes the law of dominance, law of segregation, and the law of independent assortment. I had said the law of segregation states that every individual possesses two alleles and one allele is passed to the offsprings. The 50% of genetic material from the one parenting going to be passed to the next generations. Like that, only alleles are passing from parent to the offsprings. That way, they are similar. They possess similar characters. Why? Because means this is due to the inheritance process. That means heredity. They possess similar characters. The law of independent assortment states that the inheritance of one pair of gene is independent of inheritance of another pair of a gene. That means the alleles are assorted independently without any segregations, without any closing or the mixing or the segregation of the genetic material. The alleles are passing are assorted independently. Conclusions from the Mendel's laws. I means he has conducted many experiments and he has studied whole the pea plant and has conducted pollination techniques and hybridization techniques proposed monohybrid cross experiment, dihybrid cross experiment. Based on this monohybrid cross experiment, he has proposed the law of dominance and the law of segregation. Based on the dihybrid cross experiment, he has proposed that law of independent assortment, how the individuals passes their alleles to the next generations. Here, what is the genotype, what is the phenotype, and what is the homozygous and what is the heterozygous is going to dealing now. The genetic makeup of the plant is known as genotype. I already said that genetic material of the plant is known as genotype. The genes, if in genes having the two alleles, capital T is the one gene, it's having two alleles. The that is only the responsible for the trait. That means the phenotypic expressions. On the contrary, the physical appearances of the plant is known as a phenotype. The physical characters which we are appearing with with our eye, which can see with our eye, these are called as a physical appearances of the plant. This is a phenotype. The genes are transferred from parent to the offsprings in the pairs. 50% means 50% of genetic material. In the genetic material, he is passing the genes. These genes are having the alleles. The genes are transferred from parents to offsprings in the pair are known as these are the alleles. During the gametogenesis process, that means during the formation of the gametes, when the chromosomes are halved, there is a 50% chance of one of the two alleles to fuse with other parent. Gametogenesis process, the parents possesses 50% genetic material to the offsprings. They segregating or they assorted independently, and they we can see the differences in the variations of the characters or the colors or with the different changes. When the alleles when the alleles are the same, they are also known as the homozygous alleles, and when the alleles are different, they are known as heterozygous. I discussed about that already in the starting slides. That the homozygous means same type. That means same letters will be in the. That means same genes. Genes are having same alleles. These are homozygous. If it is having different types of alleles, these are the heterozygous alleles. Thank you. We will see you in the next video.